Okay, I think we should start the class. So I'll be sharing my screen. Somebody please let me know whether the screen is visible and whether my voice is clearly audible or not. Okay, yeah, I see in the chat. Perfect. Okay, thanks a lot. Okay, so again, I welcome you all to the course Bioinformatics Algorithms and Applications. So today we'll take a look at basically the contents of uh, the the week 7 lectures by Professor Michael Gomiha from IIT Madras. And then again, we'll basically discuss some questions kind of related to the assignments that appeared in some of the earlier iteration of the course, and then we'll talk through some of the concepts that Professor Gomiha went over in his lectures as well. So in the last class, for a brief recap, so we talked about the PDB structure and its resolutions and so on, right? The different parameters that uh, can help us basically assess the quality of the PDB structure. So we sort of ended on that. Uh, and then we basically talked about some numeric questions, for example, here transfer free energies, and again some early concepts of homology modeling, which we will be kind of talking in a little bit more detail in this class. Uh, okay, so again we talked about some of the other things, for example, contact maps and the concept of basically short range or long range contact, some of that. So basically the contact between the 3D between two atoms or two residues, in this case in a 3D space. So if the distance is kind of lower than a threshold, then you can say they're forming a contact in the 3D space. But those two forming contact forming residues, whether in the sequence space they are far apart or not, so that will kind of determine whether they are forming a short range contact or long range contact, and that can give us some insight into the structural features and so on, right? Then some concepts regarding accessibility, folding, etc. And then we ended on this sort of uh, basically some statistics on the PDB database. So in this particular class, I will first talk about some of the concepts in the previous class that I did not have the chance to go through. For example, the structural uh, characterizations, or based on structural characterizations, the differentiation between some of the protein classes. So we'll talk about that in this class through like one example. Uh, and this was another question that I kind of didn't go over, I think. So for example, in aromatic sulfur interactions, the cutoff distance should be around like 5.3 angstrom. So based on these different kind of interactions, so for example, hydrogen bonding interactions, it could be sulfur pi interactions and so on. So what sort of criteria establishes that this sort of interaction is kind of forming? So it is also important to know that, so that we can verify in our actual computational data later on that whether these sort of interactions are forming between two residues or one residue and one ligand atom and so on. Okay, so the concepts regarding these distances are also kind of important. So yeah, the structural basically classification that I was just talking about. So for example, here the question is asking like what sort of databases are present which provide structural information on the which provide information or characterization on the proteins based on the structural classes. So does anybody remember some name of those databases? So I think I see in the answer Chimera. So Chimera it's a visualization tool that can allow us to sort of see the 3D structure and the conformations that are present in the proteins, and can also allow us to analyze uh, basically different parameters and so on. But here, what the question is basically asking, it's asking about the two kind of databases that Professor Komiha also mentioned. So SCOP and CAT, basically structural classification of protein database, and then class architecture topology homology. So these two databases sort of have information on the structural class of proteins. Now, what kind of classes are you talking about here? So, based on the prominence of sort of the secondary structures that are present, and you can say in some cases that the patterns of secondary structures that are forming within that protein. So, based on those, basically the proteins can be segregated into different classes. For example, in this question, it is asking uh, if a protein or proteins that contain alternate alpha helices and beta strands, which are segregated from each other. Now that is very important. So alpha helices and beta strands are present in the protein in an alternate manner. However, there seems to be some segregation between those as well. So the segregation will just look visually just in the next slide. But can somebody mention what sort of class these proteins can belong to? So of the four options,
C, alpha plus beta.
yeah. So basically, in this case, the answer will be C, alpha plus beta. So the all alpha classes is basically, if we look into this sort of diagram here, so the all alpha classes will be mainly alpha helix dominant, right? So mainly the presence of alpha helix can be seen in that in those classes. And again, all beta classes, of course, will be beta sheet dominant, right? Now, the confusion might arise in some cases. What is the differentiation between the alpha plus beta and alpha pi beta or alpha slash beta classes? So you can take a look at that. So here, basically, alpha by beta or alpha slash beta proteins, we can see. So in the right hand side picture, if you notice the sort of arrangement of the secondary structures, we'll see mostly one alpha helix is sort of adjacent to another beta sheet, which is then preceded by another alpha helix, and then after that another beta sheet is present, and so on, right? So the alpha helix and beta sheets are mainly sort of present in like one after another like that, and there does not seem to be much segregation in this case, although in some cases we see, for example, here two beta sheets are present together, but that is not much basically. If we compare that with the alpha plus beta proteins, we will see that yes, alpha helix and beta sheets are present in alternate manner. However, you see at some points, basically a lot of beta sheets are present at the like just one after another, right? So, for example, here we are seeing that the alpha helices, so there are two alpha helices, one after another in this particular position, and then after that, four alpha helices, they're kind of clumped together, we see, and in between those, we see three beta sheets just present one after another, right? So that means that there seems to be some sort of segregation between the alpha helix and beta sheet clusters or groups, you can say, in this particular case. So those types of proteins where the alpha helix and beta sheets are in alternate manner. However, there is also a segregation. So a lot of alpha helices are present like as a group at some point in the sequence space, if you notice. So those can be classified under the alpha plus beta proteins. And those where there is no such segregation can be observed, those are kind of classified under the alpha by beta or alpha slash beta proteins. So that was kind of the concept behind the structural classification based on the uh, secondary structure prominence and the pattern of presence of the secondary structures, if you will. Okay, so I have taken these sort of figures from this particular reference. You may take a look at that later.
I still didn't get what is the difference between alpha plus beta and alpha beta.
Alpha plus beta and alpha by beta. So basically, in alpha plus beta, you will notice that a lot of alpha helices and beta sheets, they are sort of clumped together. So for example, let's say residue, let's say 1 to 10, 1 to 10 is a bit too much. So let's say residue 1 to 7, that is forming a beta sheet, right? And then let's say residue 7 to 14 is forming another beta sheet. And then let's say residue after that, let's say certain a small loop is present. So unstructured loop region is present. And after that, you see another beta sheet. So that is three beta sheets that are present together just one after another. Maybe they are just segregated by a small loops, right? Loop-like structures. And then after that, after those three beta sheets, when they're ending, you will see a lot of alpha helices are present. For example, let's say residue 30 to let's say 38, that is forming another alpha helix. And then after that, maybe certain unstructured loop region is present. And after that, again, another alpha helix is present in the sequence space. And then after that, again, another alpha helix is present. After that, for example, let's certain loop region is present, and after that again another alpha helix is present. So basically what is happening here, uh, you are seeing, of course, alternating alpha helices and beta sheets, but at the same time you are seeing multiple alpha helices and multiple beta sheets are present at certain positions, just one after another, you can say, which like they're grouped together, you can maybe think in that way. So those sort of segregations, if you notice, basically then you can classify them under alpha plus beta proteins. So basically there is alternate pattern in alpha helices and beta sheets. However, these alternate patterns does not involve single alpha helix and then single beta sheet and then single alpha helix and then single beta sheet and so on. It does not follow like that. What happens is that multiple alpha helices and multiple beta sheets, those are kind of alternating between one another. So that will be classified under alpha plus beta. Uh, and then here alpha by beta, that will be again kind of like single alpha helix, uh, which are present after another single beta sheet, after another alpha helix, and so on. So basically, single units, you may think of it like that. So single units mostly alpha helices and beta sheets alternating between one another in the sequence space. So that will be sort of the alpha by beta protein. Hopefully that was clear.
So just a small doubt. So for example, in this alpha plus beta in your diagram, so this TLS you mentioned, no?
Yeah.
Four beta sheets.
Yes.
So
Yes.
these four sheets will not be considered as single beta matlab, unit with four sheets, matlab, four parallel or four anti-parallel.
Yeah.
Is it not that?
Yeah. Basically, four parallel or four anti-parallel beta sheets, you can consider it like that. So maybe these beta sheets are not like present at a single stretch, but for example, there is some they are kind of separated by like a small loop-like structures. You can see maybe that
Maybe
sort of,
turn.
yeah, turn or loops. These kind of structures are present between those. So that's how these individual units are kind of separated from one another. You can think of it maybe that way. So in a lot of proteins, you will see these sort of structures. I mean, for example, beta sheets are present one after another in like anti-parallel way, which are connected by certain loop regions. So in a lot of proteins, you can see those kind of structures, and which is kind of basically being meant here. So just like a lot of individual beta sheets, they are kind of grouped together, or beta strands kind of grouped together. Uh, so that is kind of being indicated here, basically. So here, for example, this the second point kind of mentions that alpha plus beta is like a mix of all alpha and all beta. So basically, all alpha is mostly alpha dominant, and then if you think about it, okay, that means that in all alpha, of course, the alpha helices are present like kind of clumped together at particular points, right? Because they will be mostly dominant in alpha helices. So all alpha and all beta, if you sort of kind of make an alternate pattern between those two sort of structures or structural groups, then you can get something like alpha plus beta. So maybe that's the best way. That's one way to probably look at that. Okay. Uh, okay. So yeah, we talked about the occupancy concept in the earlier classes when we just looked into the PDB structure, right? So basically, occupancy is essentially indicating that for certain atoms, some alternate conformations or alternate coordinates are captured. So in the whole crystal, for example, some molecules, they are kind of having one particular conformation for this atom or for this set of atoms, you can say. And then in some other crystallized molecules, you will see that these sort of coordinates are slightly shifted. So then if you calculate the occupancy between all of those, so it will sum up to be one basically in total, right? So here, for example, this question was asking that if the atoms of a particular residue have an occupancy of less than one, then what basically it should imply. So in this case, the best answer will be that the residue is said to assume alternate conformations. So these alternate conformations are sort of captured in as the coordinates there in the PDB structures, and it is present in the occupancy column. So the occupancy, if you sum it up all together, it should get the total of one. Okay, so yeah, so In the classes also, Professor Bromley, in multiple contexts, he went over the concept of entropy. Now, the concept of entropy is very interesting. It is also, a very good driving force of protein folding, you can say, in a solvated space or solvated system. The concept of entropy comes very handy in basically calculating the total binding free energy. So in case of drug protein interactions or protein protein interactions, it is also sort of a very important concept to go over. So here, for example, the question is asking, it's kind of a basic entropy related concept. So if you compare a solid, a liquid, and a gas, which of them should have the minimum entropy? Whether all of them should have the same entropy, or some uh, form could have higher or lower sort of entropy. Yeah, so I see in the chat solid.
Solids.
Yes. So solids should have the minimum entropy. Now, what is entropy? Entropy is basically the measure of disorder, right? The disordered nature of the system. And it kind of indicates the energy that is unavailable to do work. Now, since it is the measure of disorder, so of course we know that the solid state. So in the solid state, the molecules basically have the lowest sort of degrees of freedom. They cannot move around much, right? Uh, so that is why the solid state will have the lowest sort of entropy. And then if you sort of transform that into a liquid state, you will see that the degrees of freedom kind of increases. The molecules are kind of more free in a sense, so that they can move around, basically like the liquid or the dynamic flow of the fluids. You can maybe think of it like that. And then of course, if you transform that again into a gaseous state, so of course the disorderliness will increase further, because now it is now the molecules are basically more free in a sense, right? So solids will have the least entropy, followed by liquids, and the gaseous states will have the sort of highest disorder or the highest entropy. And here, for example, the delta S, or the change in entropy, is kind of mentioned. So in any cases, if you try to measure the change, you kind of have to measure the final versus the initial value. So from solid to liquid transformation, in this case, of course, the final entropy minus initial entropy, if you do that, then you will see the delta S will be greater than zero. And the inverse will be the case for liquid to solid transformation. And then here again, from liquid to gas, you will see delta S will be greater than zero. From gas to liquid, you will see delta S less than zero. Now, does anybody know how this concept of entropy, or how the term of entropy is sort of included in the binding free energy calculations? So we talk about binding free energy calculations being an indication of sort of the affinity between, let's say, protein and a ligand, right? And if the binding free energy, for example, is termed as delta G bind in a lot of cases. So does anybody know what sort of components make up the binding free energy as a whole? So in the formula, it basically has two other components that sort of indicate delta G as a whole. Does anybody remember what that formula was? So you look at binding free energy calculations maybe in some classes when you talk about protein drug interactions, and I think I also have one slide in this particular set of slides also. So if you remember, the formula was delta G, which is the binding free energy, equals delta H minus T delta S. Okay, so this delta H part, that is sort of the enthalpy or the change in enthalpy, you can say, from, of course, the initial state of it will be final state minus initial state. So the final state will basically mean, if you are talking about protein drug interactions, the final state will be the combined or the complex form of protein and ligand as a whole when they're bound together, and the initial state will be when the protein and ligand were separated. So that will indicate the initial state. And similarly, as you calculate enthalpy, you also have to take into consideration the entropy change as well. So here the delta S, or the total T delta S term, which is multiplied by the temperature value here in Kelvin. So this T delta S term also needs to be calculated, which will basically give you the entropy contribution to this binding. And if you combine them together, you can get the actual binding free energy. So we'll just take a look at the like one of the, for example, numeric problems related to this. Uh, so other than binding free energy, we also talked about entropy having some importance in protein folding. Okay, so we'll look at it maybe in another question. So let's try to solve another question, another one of those. So I think similar sort of questions were given in assignments in the past also. So this question is basically asking, so it is a numeric question, and you have been given, for example, 4 atoms of a particular amino acid residue. Only 4 atoms you need to consider here. And for all of these atoms, for example, here the N, the C alpha or Ca in this case is indicating the C and O for this glycine 58. So that for basically for each of these 4 atoms, we have been provided the X, Y, and Z coordinate values. And then you have also been provided the atomic mass for these atoms. So for carbon, nitrogen, and oxygen, it is 12, 14, and 16 respectively. And then you have been asked, okay, so what should be the center of mass of glycine? If you sort of try to calculate it from these set of coordinates. So can somebody maybe try to calculate it and share in the chat? So the formula for calculating this sort of numeric questions in the case of center of mass will be something like this. So in this case, of course, on the numerator, you see summation of mi times xi. So this xi, or conversely, if you consider the y and z coordinates, it will be yi and zi and so on. So you have to calculate for each atom, basically consider their mass, consider their coordinate values. You have to do that, and then you have to do a summation of all of that, and you can divide that by the total sort of summation of masses for all of these atoms, and then you can arrive at the conclusion. So we'll get in this case three different calculations. Basically, one will be for the x coordinate, one will be for the y coordinate, one will be for the z coordinate. So for each of these coordinates, we have to sort of calculate the mi times xi. For each of these coordinates, for each of these atoms, you have to calculate the mi times xi and divide that by the summation of mi, and then you will get the answer. So we will try to calculate this and share in the chat. So for example, if we just take a look at the first coordinate value, so for the x coordinate, what you will have to do is basically you have to take all of these atoms, and for each of these cases, you have to take their x coordinate and their mass, basically atomic mass, and you have to multiply that. So for example, the first one, so the first atom is nitrogen, of course. I see one hand. Yeah, please go ahead. Okay. So if you have any questions, you can also share in the chat. I'll try to take a look at it. So in this case, in this particular case, for example, the nitrogen, we have been given that nitrogen's atomic mass is 14, right? So the first term for the mi times xi will be nitrogen's atomic mass, which is 14 multiplied by 6.3, right? 14 times 6.3. And then after that, for carbon, we have been given the atomic mass as 12. So 12 times its x coordinate, it will be 12 times 6.1. and then plus another carbon, so which is 12 times the x coordinate value 7.4, so 12 times 7.4, and plus you have to do for the oxygen also, which has an atomic mass of 16, so 16 times 7.4, and then you divide that by basically the sum of all of their masses, atomic masses, right? So we have basically 2 carbons, 1 nitrogen and 1 oxygen. So you sum all of that up, and that will be your denominator. And you do the calculation, you will get 6.83 here. So can somebody maybe share what the answer could be? It should be something something between A and D, right? So I think I see one answer D in the chat. Yeah, so if you sort of do the calculation for the y coordinate as well, and if you do the calculation for the z coordinate as well, so you'll see the value 6.83, 3.04, which is kind of close to 3.00, and the 9.003, so 9.0 here. So in this case, the answer will be D, as I see in the chat, multiple people have said. Okay, so if you get sort of these sort of numeric questions, then you can apply this formula to reach the answer, basically, right? Now, one of the concepts, one of the kind of main concepts in these week 7 series of lectures, it was basically protein modeling, right? Structure modeling of protein. And the concept that we, for example, got to know, the main component of protein modeling, one of the main components, so it was called homology modeling, right? One of the main processes through which the modeling is done. And in particular, for homology modeling, so there is sort of this concept between, let's say, number of aligned residues. So in homology modeling, you have to do sequence alignment. And then if you plot sort of the number of aligned residues and the percentage of identical residues, then you will kind of get a plot kind of looks like this, where the safe homology modeling zone, where the values are sort of ranging in this space, this upper space. So you can take for any alignment corresponding to that identical residue. So those type of alignments and those kind of hits you can take as your initiation for the homology modeling. However, there is another zone kind of which is called the twilight zone, which is different from the safe homology modeling zone. So that is what this question was asking. So for 100 residues, if 100 residues are aligned with 30% sequence identity, you will see it sort of falls like around this region probably. So it does not go to the exact safe homology modeling zone. It will be kind of part of the twilight zone. So these sort of questions might appear also. We'll take a look at the processes of the homology modeling. So I think Professor Bhumiha went over the concepts, and then he also used one software called Modeler in the demonstration for the homology modeling. So we'll take a look at some server-based basically programs such as Swiss model that can do the homology modeling for us. And using Modeler, we'll also take a look at loop modeling in particular. So in a lot of protein structures, crystallized structures from the PDB database, we'll see that there's a lot of residues have their coordinates captured, but in certain unstructured and or loop regions, where due to high flexibility, their coordinates were not been able to capture properly. So those sort of residues you have to model in some way. So we can use again softwares like Modeler to model those missing loop regions, which can give us a proper structure to start our further in silico processes. Uh, so another kind of concept that uh, basically was sort of part of this entire protein folding related concept, protein modeling related concept. So that is, of course, the calculations between, for example, the unfolded state, the energy terms related to those, and then when the proteins, for example, sort of convert from the unfolded states to a folded states. So what sort of energetic change is going on there? Can you calculate those energy changes and so on? And in this case, the question is asking kind of a more basic form of that. So it is asking what sort of forces drive the folding of protein. We just discussed about it in the earlier classes, and in the earlier slides I also talked about it. So from the unfolded state of a protein, when the protein folds into a 3D structure, of course in a solvated space, we notice, I think I see some answers in the chat. So these sort of transformation is mainly driven by entropy. Okay. Now, why is that? So basically, I think we discussed in some earlier classes also. So let's say you kind of drop a protein sequence or just the primary sort of structure of protein in a solvated medium, right? So let's say here the sort of the chain is kind of looking like this, right? And for each amino acid components, uh, I am kind of representing that by the dots here in the middle. Now, what is happening is that water naturally has like a very good hydrogen bonding pattern that is formed between the different water molecules, right? Now, when you drop the primary structure of protein into the solvated medium, what happens is that due to sort of the disruption in the hydrogen bonding patterns between the water molecules, the waters tend to form a highly structured solvation layer around the amino acids that are being dropped there. Okay, so this sort of uh, bubble that you are seeing around these dots, so these are kind of, you can say, the solvation layers, right? Now, of course, these solvation layers are very highly structured or very highly ordered, you can say. Uh, so now if we sort of take a look at what happens during protein folding, so that might give us an idea about how the entropy change is basically driving this process. So when the protein is folding into 3D structures, you will see that the amino acids are present in a much closer sort of space than when they are sort of forming the primary structure just like that, right? So when the clumping of the amino acids occur, you will see the collective volume of the solvation layer or the surface area that the solvation layer covers around this amino acid molecules clumped together, amino acid residues clumped together, which is sort of the 3D fold of the protein. So you'll notice that if you sum up all of the individual uh, basically surface or volume that is covered by the solvation layer in the left hand side, so you'll notice that the sum of all of these individuals is reduced after the amino acids are clumped together and a big sort of solvation layer is formed around them. So basically what is happening, since the solvation layer and the volume or surface that it covers, it sort of represents a highly ordered state, a highly ordered structure of waters. If the collective surface or the total volume of the solvation layer decreases after the basically hydrophobic clumping or the amino acids kind of get clumped together, so that will basically subsequently indicate that the disorderedness of the protein has decreased, because earlier uh, the solvation layers, the total sum of all of their kind of covered surface or volume was higher, and that basically indicates a higher value of the ordered structures or ordered system being present here. And then when the amino acids get clumped together, the total or cumulative surface or volume that is covered by the entirety of the solvation layer, so that total value gets decreased. So that means that the value of the ordered structure or the orderedness, that will also get decreased, and that can basically indicate okay, so that means the disorder will increase slightly, if you think of it that way. So that means the entropy will be higher in this case, in this transformation from this primary structure to this tertiary structure transformation. And due to this entropy kind of favored process, entropy favored basically transformation process, you will see that the hydrophobic amino acids kind of tend to clump together. So uh, that will indicate that is sort of what is meant by the hydrophobic clustering, and it is driven primarily by this entropy related process. The other sort of fundamental forces, for example, van der Waals forces or charge Coulombic interactions or electrostatic interactions, those primarily take part, or hydrogen bonds, those take part in uh, basically stabilizing the 3D fold. You can think of it in a way, but we cannot say that they are directly responsible for the 3D fold, but they take part in stabilizing the entire 3D fold. So that was sort of the concept between this entropy driven process. Yeah, now the basically inclusion of the entropy terms. Yeah, I see another, I think, hand in the chat. Yeah, you can.
Sir, am I audible?
Yeah. Please, please.
Please.
Yeah,
One
yeah.
second, sir.
Huh.
Sir, I did not understand last question. You said entropy is like it's just a disorderness, right? How can it be dominant something?
So basically, the law of thermodynamics states that uh, for a spontaneous process, the entropy of the universe increases, right? So that means if a system is sort of being transformed from a state where the disorderedness was lower, or it had sort of a relatively lower entropy, to a more highly disordered sort of state, or a state with a higher entropy. So that transformation will be favored in this case. Now here from just calculation, so basically since the solvation layers indicate a highly ordered structures, and orderedness is basically the opposite of entropy, right? So then we can think of it in a way that, okay, so if the solvation layer is indicative of the ordered structures, so if the total volume or surface covered by the solvation layer decreases, then we can say the system is moving more towards a disordered state uh, uh, in terms of basically entropy in this kind of context here. So since in this particular case, the cumulative volume when the hydrophobic amino acids are clumped together in a say or in a protein tertiary structure, the total solvation layer that is formed around those basically amino acid residues or the protein folded protein as a whole. So the surface or the volume covered by that will be lower than the sum total of the individual solvation layers that are forming in the left hand side state. So that will kind of indicate that, okay, in a solvated system or in a solvated state, moving from the let's say just a primary structure of the amino acid residues chained together to a folded state where the amino acids are grouped together. The hydrophobic amino acids are forming the core and the hydrophilic are on the surface, but they are grouped together. So this transformation will be favorable because it leads to an increase in entropy. An increase in entropy is kind of favorable by the laws of thermodynamics. Okay, so that is why it is stated that it is entropy driven, uh, because basically the increase in entropy or the increase in disorder from the primary structure to a folded state, that dictates the folding. So that was what the question was asking basically.
Okay, sir, I got it.
Yeah. Okay. Now, what is the inclusion of the entropy concept in the binding free energy calculation, for example, in protein drug interaction, protein interaction also? So we saw the formula earlier, right? Now here we have a chance to basically apply that and calculate one numeric problem. So here, the data that I'm showing you, it is basically from a molecular dynamics simulation and subsequent free energy calculations, right? So here we had one protein and one ligand molecule bound together, so a protein ligand complex. We simulated that. It achieved equilibrium. We simulated for something like 200, 300 nanoseconds, right? And then based on the equilibrated trajectory, we took snapshots, you can say, uh, we took like a part of the trajectory where the system achieved equilibrium, and we applied that or used that uh, to calculate the binding free energy through a particular protocol which is called MMPPSA. So you might know from a lot of like in silico drug discovery or basically in silico protein interaction or similar kind of research papers. So this MMPPSA protocol is widely used in calculating the binding free energies from molecular dynamics trajectories. And similarly, you might have also heard about another basically protocol which is called MMGBSA. So MMPPSA stands for molecular mechanics Poisson Boltzmann surface area, and then MMGBSA stands for molecular mechanics generalized Born surface area. So GBSA is kind of an approximation of PBSA. But in this case, we applied the MMPBSA protocol and as the result, as the output, we have gotten this sort of results. Okay, so here what the data is saying basically, firstly you can see on the upper block, it says differences, so complex minus receptor minus ligand. Now we looked at earlier that the binding free energy was basically delta G, right? So delta will indicate the difference or the change from one state to another state, and in this case the delta G or each of the components of the delta G, when those will be calculated. So first you have to calculate the final state value, which will be the complex or the bound state value, minus the individual values for the protein and the ligand, or the receptor and the ligand. So that is why the difference is mentioned here, complex minus receptor minus ligand. And if you sort of, so the sum total is termed as the delta total in this case. If you look at the individual components here, you will notice that this delta component is formed by these sort of like individual parts, right? So can somebody mention what these energy terms are indicating? What sort of interaction related energy terms are being mentioned here? So you'll notice first of all, a average value, and then you'll also notice the related standard deviation and standard error of mean in this case also, right? So basically, since this is calculated from a trajectory, so trajectory means a collection of snapshots. So for each individual snapshots, the values will be calculated, and then you have to take an average of that. So that the program does for us, and then it reports the average value as well as the associated standard deviation of, and standard error of mean also. So here the terms are basically firstly van der Waals. So the van der Waals energy term. Then you have the this EEL, which is indicating the electrostatic interaction energy term. Then you have this polar EPB, polar solvation energy term. You have the non-polar solvation energy term, and you have the delta total, which is sort of, if you sum all of this together, you will get this value. Two other terms are also mentioned here, this delta G gas and then delta G sol. So delta G gas is basically the sum of van der Waals and electrostatic, and the delta G sol is the total solvation free energy, which is in this particular case the polar solvation and the non-polar solvation. So if you look into sort of the exact process or the protocol that these sort of algorithms follow, then you will notice that the solvation is sort of calculated in a way where the polar and non-polar are done kind of separately. So it is kind of a thermodynamic cycle where if you kind of add up the individual components, you will get this sort of values, right? Now here, of course, the main thing is this van der Waals electrostatic polar and non-polar energies, and then the sum delta total, right? On the bottom block, you will see it sort of mentions the entropy term. So if we remember the formula, so it was delta G equals delta H minus T delta S, right? So this T delta S particularly, that is being indicated by this entropy term calculation. Okay. And here again, difference is of course complex minus receptor minus ligand is being calculated. Here we see the value of minus 17.8647 for the entropy term. So for the entirety of the T delta S, and for the delta total on the upper hand side, so that is minus 31.4008. So this delta total is basically delta H. Okay. So can anybody mention what will be the binding free energy value for these data set, for this trajectory? So I see one answer C. Others can also try maybe. Yeah, so basically, okay, so in this case, all units were in kcal per mole, and the temperature was 298.15 Kelvin. But yeah, basically, if you use the formula here, so let me just erase this one. Yeah. So basically, if you sort of use this formula here, you will see, of course, the delta H value is minus 31, and then the T delta S is minus 17.8647. So if you do a minus of that, it will be plus 17.8647. In total, you will get minus 13.5361 value. Okay. So hopefully the math is correct here. So in this case, you should get the result A, right? Minus 13.54 if you just add all of this total, add all of this together. Now, of course, one question is kind of imminent here. So what does this minus sign indicate? So it is the value of minus 13.5361, right? So is there any significance to this minus value in a calculated
Favorable reaction.
Yes. So since you are calculating the value for the final state minus the initial state, so a minus term basically indicates that the final energy value or the energy term for the complex that was lower than the initial protein and ligand combined together, right? So that means during this transformation from the initial to final state, from the unbound to the bound state, some energy was liberated outside. So that is what is being indicated by this minus sign here, minus symbol here. So since energy is being liberated outside, so that means the entire system is becoming more stable, and more stable is better in terms of binding affinity, of course, because we want our drug molecule to stably bound or stably occupy the binding region of the protein. So that is good, right? Let's say we had this minus 13.5361 kcal per mole. Let's say this was our compound 1, right? Let's say we had a control compound or a control ligand, which we use to compare our novel drug molecules, right? So let's say we had the controls calculated binding free energy value as something like this. Let's say minus 9.85. Okay, kcal per mole. Let's say another compound, comp2, it had the value of let's say minus 8.7 kcal per mole. Okay. So we have compound 1 minus 13.53, control minus 9.85, comp 2 minus 8.7. So can you say which new compound between compound 1 and 2 should we propose as sort of a better drug with a better binding affinity? Which one can we propose as a better drug?
One.
Yeah. So compound 1, since it has a more negative value, minus 13, as compared to the control minus 9.85, so you can maybe think of it, okay, so this compound 1 could be having more, higher affinity, so it is binding more strongly to the binding pocket. So you can maybe propose that as a novel drug candidate. Compound 2, in this case, since there is already the control is having sort of more affinity or better energy values than the compound 2, so you cannot basically introduce it as a new candidate, as a alternate to this control molecule in a way, right? Of course, that is we are only proposing based on the binding energy values. And of course, if it is an in silico paper, so there might be some sort of associated admit properties calculated, which sort of assesses the physiological behavior of this drug in the physiological systems, right? Through some prediction models. But it has to go through all of these processes, all of the processes that follow the computational protocols. For example, your wet lab studies, then your in vivo studies in mouse models and so on, the preclinical trials, clinical trials, and so on. So those processes need to be followed, and if it passes all of these processes, then only all of this, this entire setup pipeline, then only you can say that, okay, we have got a new drug molecule, a novel drug candidate. Before that, you cannot say, but based on binding free energy calculations, uh, based on the in silico protocols, for example, virtual screening, docking, and subsequent AMD simulations, so you can use that to, let's say, more quickly propose a novel hit candidate, which reduces the total time in drug discovery and the total cost in drug discovery by a lot. rather than if you just were to pick up a random molecule and then proceed for, let's say, wet lab experiments and start from there, right? So it is helpful in that regard. Uh, so here one question we also had in some of the earlier classes, the earlier iteration of these courses. So for example, NH at the dot 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 O, that is a hydrogen bond being formed, let's say, between some protein ligands. So what is the hydrogen bond donor and what is the hydrogen bond acceptor, if I will?
So O is a hydrogen bond acceptor. Sorry,
Yeah.
O is a hydrogen bond acceptor and N is donor.
Yeah, O is the hydrogen bond acceptor. So here in one of the earlier iteration of the courses, they provided the hydrogen bond donor as N. I guess because the hydrogen was bound to this nitrogen here. So I guess they are considering the nitrogen basically as the donor here. But it is kind of confusing between this one and this one. Kind of what should be followed. But anyway. So yeah, O is the acceptor and N is the donor here, right? So if we take a quick look at the hydrogen bond, so it is basically sort of a dipole-dipole attraction that is observed between molecules, right? It is characterized by the attractive force between a hydrogen atom which is covalently bonded to a highly electronegative atom and another electronegative atom. So basically what is happening is that if hydrogen is bound to an electronegative atom, so due to the electronegative atom kind of pulling the electron cloud towards itself, so there will be a partial charge distribution over these particular atoms, right? So for example, nitrogen and hydrogen, between nitrogen and hydrogen, you will see that nitrogen will pull the electron cloud towards itself due to its high electronegativity, and you will see a sort of delta minus over nitrogen, right? A partial negative charge around this nitrogen. And conversely, of course, you will see since the electron cloud was pulled away from the hydrogen, so there will be a partial positive charge, like delta plus is indicating that here, right? And then if another electronegative atom is somewhere near, so what could happen is that these hydrogens can form sort of an attractive, through an attractive force, sort of a bonding between another electronegative atom. So this del plus will form a bond between another electronegative atom, and that will be the basis of the hydrogen bond, right? So here are several concepts. Yeah, please go ahead. I see one hand.
So is NH a hydrogen bond donor or N hydrogen bond donor in the previous example?
Yeah, so in one of the earlier assignments for some of the earlier years, so they said as N being the hydrogen bond donor. But it is kind of confusing because you can think of NH as a total group in a sense. But since I think their reasoning was that since nitrogen is sort of donating the hydrogen, because initially the hydrogen was attached to the nitrogen, and then the donating is being done by the nitrogen particularly. So they sort of said that nitrogen should be the answer in their, I think, assignment answers. Okay.
Okay,
Yeah.
but
So
when we go through the, if you go through the various books, then it is always mentioned as NH. So I was a bit confused.
yeah, it is
Okay.
kind of confusing. So maybe the
Yeah.
proper answer should be NH, but in this course's answers earlier, it kind of had been mentioned as N. So this is kind of
Okay.
confusing in that regard. Yeah, that is
Yeah.
true.
Okay. Okay. Thank you.
Mhm. Yeah, so basically the electronegativity discrepancy kind of leads to these bonding, right? And here it is again talking about the increasing electronegativity trend for these different atoms, for example, H, N, O, F, and that will sort of be part of the strength of the hydrogen bonding as well. Here, another interesting thing is mentioned here. So the bonding energy of the hydrogen bonds, it is sort of in the range of 8 kilojoule to 50 kilojoule per mole. So 8 to 50 kilojoule per mole. You can convert that to kcal per mole by dividing with 4.2. So it is not very strong, of course, not as strong as the covalent bonds, but it is still an important part of let's say non-covalent interactions, or when we analyze the protein ligand sort of interactions, what different interactions are going on there, the hydrophobic interactions, the hydrogen bonds, maybe the pi-pi stacking and so on. So there we also have to analyze how many hydrogen bonds are being formed and what is basically the source of the binding affinity that this particular ligand molecule is showing against this target protein. So there the hydrogen bond analysis also needs to be done in a lot of cases. Okay. Yeah. So now we come to one of the main concepts of the week 7 series of lectures, the homology modeling concepts, right? So for the homology modeling, we know that it is sort of a predictor of the structure, three-dimensional structure of a particular protein molecule based on sequence homologs, right? So if another sort of structure or a set of structures are present already in the PDB databases, which if we compare their sequences, they will sort of be homologous to this unknown structured protein. So we can use their 3D structures and you can apply those in our new sequence or the unknown sequence whose structure is unknown. And then we can try to predict the 3D conformations of the particular amino acid residues, their side chain orientations, and so on. Their whole like backbone formations, in what structure the backbone is kind of being folded. So you can try to compute that through this process of homology modeling. So here the question is asking that if in homology modeling, if the template and the target residues are identical, so you have a template, you have found a template, a proper template for the modeling. You have aligned that sequence with the target sequence, right? And then you are seeing at some positions, of course, the same residues are present in the template sequence, in the target sequence as well, right? So then what you can follow as a protocol? Can you copy all of the main chain and side chain atom coordinates for your prediction? Can you only follow the main chain atoms but not the side chain atoms? Can you can you copy the side chain atoms only for the prediction? So this is the kind of question this one is asking. So from option A to T, so basically it is asking in a combination of like what can you do? Can you copy the main chain coordinates? Can you copy the side chain coordinates? Can you copy all of them? So any idea what this is saying? What the answer should be here? So through the homology modeling process, we will see for the backbone generation or the backbone calculation, the backbone structure calculation, of course, you need to copy the main chain, right? In this particular case, it is saying that the residues for which you are trying to predict, in that sense, in the template and the target, the residues are identical. So in the alignment, there is not a mismatch in that position. There is no gap in that position, but the exact residue is present in both cases, right? So in that sense, what you can do is you can copy the main chain and side chain atoms for the same amino acid residues. Okay, so for the amino acid residues or positions where the sequence is matching exactly, for those particular positions, for those points, you can copy the coordinate information for the main chain as well as the side chain from the template, and you can get the values for the target. So here in this case, the proper answer should be option B. So option A basically indicates that all main chain and side chain atoms coordinates are copied. So all is kind of confusing here. So not all of the basically residue values you can copy, because of course there will be some mismatches and so on. But for the ones that are exactly the same, for the ones that are matching perfectly, for those you can copy the main chain and the side chain atoms. Okay. For the other ones where you don't see a match, so in that cases you have to only copy the main chain atom coordinates. And for the cases where the alignments are indicating a gap being present in some places, so to fill up those gaps, you have to do some sort of loop modeling, right? So you have to sort of generate the conformational orientations, and you have to minimize those and so on. So that is part of loop modeling basically. If we take a look at the process, so basically homology modeling is also known as comparative modeling. And of course, it predicts the protein structures by leveraging sequence homology with established structures. So if you have homology with already known protein structures, if you have homology with the sequences, then you can use that to predict the structure of the new target, right? Yeah, and it basically is some other concepts are being told here. For example, significant sequence similarity, if there is present, then the assumption is that they will likely exhibit comparable three-dimensional structures, because of course the importance of sequence is definitely there, right, in the 3D folding. So if two sequences have high sequence identity, let's say 90%, 95% identity, then of course their 3D fold will be a lot similar, right? And then if one protein sequence is known, homology modeling could confidently transfer their structures into another unknown protein structure, basically sequence to predict another unknown protein structure. So it is generated through all atom models, and then various steps are there. So if you look at this diagram in this flow chart, you will see firstly you have to identify a target sequence. So for the target sequence, ideally you will have a very high sequence identity. So that will basically make your model more accurate. Then you have to do so I was talking about the template sequence, sorry. So for the target sequence, of course, you have the target sequence. Then after you do the database search from a number of templates, which will be given as hits, you have to sort of identify one particular template. So you can take a look at, let's say, high sequence identity, then corresponding to that template, so the template structure. So if any missing regions are in those structures, so you can sort of try to eliminate that. You can try to find a new one, a better one maybe. So balancing all of that, balancing the presence of missing structures, balancing the basically the sequence identity and all of these things. Then you have to identify proper template through that. And then you have to do refinement of the sequence alignment. You have to build the model framework. You have to add the loops and the side chains. And then ultimately you have to optimize the model and then evaluate the model. Basically, by going through or by placing the structure through a number of parameters. For example, in Ramachandran plot, whether in any residues are showing or falling into the area of unallowed regions and so on. So these sort of evaluation process you have to also follow. If you take a look at the individual parts, for example, firstly the template selection is coming. So it is very crucial because proper template will lead to a good structure basically. So it is the first step. Foundation for the entire modeling process, where you basically search the PDB database for homologous proteins with already known structures and hopefully structures with high resolution. So that will also be quite good, right? Then you have to do some sort of pairwise alignment using tools like BLAST or FASTA. And basically here it is saying that at least 30% sequence identity should be present with the query sequence as the template. And then 25% in some cases can be acceptable maybe. Now, if multiple structurally similar database proteins are identified, then you have to choose the one with the highest percentage identity, but you have to also take into consideration the resolution, appropriate cofactors, etc. So whatever sort of other information that are present in the structures, the quality of the structure, so you have to take into consideration all of those. Okay. Now, if no highly similar sequences are found, of course, then template selection will be challenging. Then probably the side blast or the fold recognition, for example, threading and some other processes can be followed to generate the homology models. Now, sequences with distant homologs. So distant homolog means the identity will be lower, right? Then the modeling can be limited to aligned domains. So the only domains that are kind of conserved throughout the evolutionary process, which will be aligned perfectly. So then the modeling can be limited to those domains only. And for the target protein, due to, of course, local similarity will be there, because if a stretch of subsequence is matching, then it is very likely that the structure for the subsequence that could also match, right? Now, following template selection will come the sequence alignment. So you have to do full length sequence for the template and the target protein, and then you have to define using some sort of refinement algorithms in processes such as realignment. So this will assure the quality of the homology models. Correct alignment is very crucial, of course. And then if there is some error in the alignment at this stage, so that cannot be rectified in the subsequent modeling states also. So you have to properly align. You have to carefully evaluate the alignments also. So some algorithms are also being mentioned here. And basically that is your entirety of sequence alignments. And then after that, what you have to do is you have to do the backbone model building. So the protein backbone, the atoms for the backbone construction, if you notice that, so those will be the nitrogen, the C alpha, and the CO of the peptide linkage, right? So these N, C, C alpha, and O, so these basically form the backbone of the protein. Now, for after you achieve the optimal alignment, then you can think of it in a way that, okay, the target, the aligned regions of the target proteins residues, so they will adopt a similar structure to the template proteins, of course. So then what you can do is basically you can copy the coordinates for the aligned regions from the template, and you can just apply that to the target protein to find the positioning or the coordinates for the target protein in those atoms, right? Now, if the residues are identical, then what the earlier question that you solved had, both main chain and side chain atom coordinates, those can be copied. All of them can be copied. If they differ, then only copy the backbone atoms, and the side chain can be modeled later. Okay. So the side chain construction is coming after, and then what it is basically saying that some sort of template dependent concepts it is talking about. And then the choice between using the average coordinate values of the templates or selecting the best parts from each template for modeling. So that has to be done. So that could be analyzed case by case basis. But if you, for example, after doing the backbone modeling, so there will be some regions where gaps were present in the alignments, right? Now for the gaps, what we'll have to do is, since you cannot copy it particularly from the template part, so in that case, it will create a hole in the model, of course. Now this particular part necessitates the loop modeling. So it is also a challenging aspect because it takes some computational power as well. And you can model the loop regions in some ab initio way. So it has been mentioned here that two processes can be done. So firstly, the database searching method and the ab initio method. So the ab initio method is basically implying that without using any sort of templates, you just build the model from the first principles. So for the loop structures, for example, backbone, depending on different physics related force field terms, then what could be the minimized conformations of these loops. So that can be implied, or that can be used in the ab initio modeling for the loops as well here. Database searching, of course, is slightly different. So it is basically finding suitable segments from known protein structures in a database that fit into the two stem regions, the flank regions, that the loop between those two that has to be modeled, right? So it can be fit into there. So that is database searching. Another is ab initio, where you use the physics-based force fields, for example, to properly find out what could be the actual conformation, right? And then you do the energy minimization to get the appropriate ones. So that is basically it is mentioned here. So for relatively short loop regions or short missing residue regions, for example, 3 to 5 residues are 3 to 5 residues are missing, then you can apply both database searching and ab initio. Both of them can construct reasonably accurate models. If the loop becomes longer, of course, achieving the reliable model is considerably challenging. If you try to proceed for the ab initio calculation, then that will also take a lot of computational resource. So that has to be taken into consideration as well. After the main chain part is done, then what you have to do, you have to do refinement for the side chain atoms. Now, for the appropriate conformations of the side chain atoms, So that will be indicated by the possible conformations for the torsion angles. At each torsion angle, for example, it is mentioned here. Now that can be built by exploring all the possible conformations. So the entire conformation space if you are exploring, but of course, again, that will be highly computationally expensive. Uh, Nowadays, what people do, or the main basically approach that people take, is that they take the help of different rotamer libraries. So basically, these are the side chain atom torsion angles that are derived from already known protein crystal structures, and from those kind of rotamer libraries, they try to identify which uh, phi psi and the other torsion angles that these side chains can inhibit if they are present in sort of this sequence pattern. So the rotamer libraries, of course, contain the favored side chain conformations ranked by their frequency of occurrence, which ultimately reduces the computational time because you now do not have to explore all possible conformations for all of the possible torsion angles and so on. So that will be a side chain refinement using rotamer libraries. And then after, of course, you have to do the refinement. You have to do some sort of energy minimization, for example, uh, to minimize any static clashes if they are formed in the structures. And ultimately, you have to evaluate the entire basically built model. You have to go through stuff like Ramachandran plot if any residues are falling in the unfavorable part or the unallowed regions, basically. So you have to try to minimize further their structures and then see whether that improves the model or not. So that is the entirety of the process of the homology modeling in a nutshell, right? For checking the structure quality, you can take help from several servers. For example, ProCheck is one of the servers where the parameters for the models are compared with already established, well-defined high-resolution structures. And if unusual features are detected, then it will highlight those regions for further examination or refinement and so on, right? So if we take, for example, a quick look at one associated concept. So I'll just uh, share from another basically screen. Hopefully that is visible. Okay. Now, okay, hopefully my screen is visible now. So
Yes, sir.
yeah. So basically, earlier in some earlier classes, we looked at one visualization tool that allows us to see the 3D structures and perform some analysis and so on, right? That was Chimera, UCSF Chimera, right? If we, for example, use Chimera and open up the structures here for this one particular protein, so this structure, right? So it is being mentioned here as 2IQH. Okay, so you will see what particular protein it is. So you can search the 2IQH accession code here in the PDB database. And you will see it is basically for influenza A virus, the nucleoprotein NP, and it has a resolution of 3.20 angstrom, right? Of course, here you will see that multiple chains are present. In this particular case, I have only taken the chain C because for drug design, we probably don't need to consider all of these three chains since the drug is not binding at interface between multiple chains. So you can only consider particular chains, right? Now, before starting our any in silico process, of course, we have to closely examine the structure. So if you open up Chimera, if you try to take a quick look at the structure, let me just change the preset quickly. You will see in multiple regions, for example, between different uh, residues, like between two residues, the exact conformation is not given, but in its place, some dotted lines are present, right? So I am seeing one here between this threonine 396 and this serine 402. So between these two residues, we see some dotted lines being present. And then another one is also here, for example, serine 438 and alanine 428. So between these 438 and 428, so that 10 residue part is also seems to be missing here. So the dotted structures are basically indicative of your residues whose atomic coordinates could not be captured. I am seeing another part here also. So if you quickly go to the favorites tab and then open up the sequence, you will notice that throughout this sequence space that is being represented here in this chimera tab, so you will see some red rectangles in some of these places, right? So here I am seeing one red rectangle which is between aspartate 72 and threonine 92. Another one is here, threonine 396 and serine 402. Between those, we see a red rectangle. Then in alanine 428, serine 438. So between those two residues, we see another red rectangle. So these rectangles are basically indicative of these missing residues that you are seeing in the structures also, these dotted lines, right? Now, what you can do is you can go to favorites. So let me change the color of this to gray, for example, first. So what you can do is you can go to this tools tab, and then under that you will see a lot of analysis are being mentioned here. So in the structure editing tab under that, you can see one part that says, or you can see one button that says model/refined loops. So if you press that, it will basically open up another tab where you can use the modeler software to do the loop modeling, right? Now, of course, you have to separately install modeler from their particular website. Uh, After the installation, you have to provide the installation path here if you want to do the loop modeling in your local system. Also, for example, in my case, it is in this case the executable /usr/bin/mod 10.6. So that executable path needs to be given here. And of course, you have some options here. For example, model/remodel. We will only consider the non-terminal missing structures. So the rectangles that are present in between, we will not consider the terminal parts because those are anyway highly flexible and do not influence the drug binding. Basically, if the binding site is present at some port region inside, right? So the terminal regions are not influencing that. So we'll only consider the non-terminal missing residues in the middle. These three middle regions. These three basically red rectangles in the middle, right? Then it is asking for example, how many adjacent residues are allowed to move. So if you say one, so it will consider the residues just adjacent to these rectangles. For example, the aspartate 72 here, the threonine 92 here. So those will be allowed to move, and their conformation or coordinates will be changed according to the model, for example. And then how many models you are trying to generate. So it will generate the best 5 models here. It will give that output. And then the executable path, as I said. And basically that's all. You can also use some custom modeler scripts if you have. But here I will just go through the default options here and I can just press OK. You will see one sort of indication here how much the job is being completed. So it is currently saying 14% complete, right? After it has completed to 100%, hopefully on this tab that is open here. So our new structures or our build structures will be present and some sort of scores indicative of the quality of the model structures that will also be present here, right? So it takes some time to build the model. Uh, in some cases, you might also see that the modeler kind of has stopped working. And for example, in some of my earlier attempts, so what happened is that uh, the modeler was not giving me the model just outright, but in the particular folder for the path that I had given. So here the modeler basically generated a lot of structures, one, for example, template structure folder as well, and one script, basically Python script, which is this one, the modeler modeling.py. So it generated that script. Now, if some sort of script is generated and you are not getting the output outright in the open tab, what you can do is you can manually run this Python script. For example, I can just use Python 2 in this case, and then I can run the modeler modeling.py. So that will also be okay. So these sort of processes that are being run here will be indicated in the terminal as well. And then ultimately, you will get the output with the Z-top scores and the models that are being generated corresponding to the Z-top scores here. Okay, so that was another approach that you can try if you can use the Python scripts for the modeler also. Here, our job is still 14% complete, so it takes a lot of time for the model to get finished. So you can wait for that. Now, Swiss model is a homology modeling server, of course. And in this particular case, from the entire setup sequence, you can generate the structures. So if you start modeling here, it will ask you to provide the sequence in the FASTA formats. Here it is written that the formats must be FASTA, cluster, 10 string, or a valid uniprot KBAC. So what I can, for example, do, I can just go to my Open Chimera and then go to the favorite and then the sequence tab again. And for the non-terminal residue range, I can just try to copy this, do Control C, and then let's say I just paste it here. Okay. So hopefully that pasted the entire stretch. Then again, you can do is, for example, you can just keep the project title, you can add your email address, and you can start to build model. You can search for templates as well. But let's see what this build model button does. So it will open up another tab. If your job is not on queue, it will try to run it immediately. If it is on queue, it will take some time to finish. So it is saying auto model is running here. More models are still to be built for this project. When the entire process is done, you will see a lot of hits. You will see the corresponding identity scores. On the right side, you will see the structures that are being built for considering those templates. And you will see some sort of color codes that are indicating different parts and what confidence level those different residue ranges or different parts of the protein are built at. So if that has a high confidence level, then you can take those structures. That will be fine. Probably further structural minimization will be required because anyway, if you are planning for some molecular dynamic studies, then you have to equilibrate the structures following minimization processes, right? So that's okay. Here the modeling is again taking some time. So it is saying 28% complete. So I'll just let it run for some time. We'll come back here. In the meantime, we can discuss some of the other questions. So let me just stop present from this one, and then I can use the other screen. So hopefully this slide is visible again. Now, another set of numeric problems which was discussed by Professor Bromiha in the classes also. For example, in the context of fluorescent based identification of protein folding states, you can say. So the basically the ratio of the unfolded versus folded proteins through the urea denaturation experiment. So these sort of concepts was given. I think this exact problem was kind of mentioned by Professor Gomiha. So it is asking that in a urea denaturation experiment, the fluorescent intensity for the initial folded state was 366. Now, in a completely unfolded state, we see that the fluorescence intensity is changing, so it is 51. Now, the question is asking if you are introduced a certain molar of urea, of course, that will lead to denaturation, right? Urea-based denaturation. Then at some point, you are getting a fluorescent intensity of 256. So the question was basically asking, in that particular state for which you are getting the fluorescent intensity of 256, what is the fraction of unfolded protein as compared with reference to the folded protein? Okay. And then it is further asking what will be the free energy change for the transformation. So for free energy change calculations, we know some sort of formula that we discussed earlier also. So for example, minus RT ln K. So in this K, you can substitute it by any sort of fraction, which will give you sort of a free energy change value. in energy terms, right? So we used that. I think in one of the earlier classes we talked about the free energy landscape of the free energy or the free energy surface of protein uh, based on the populated conformations at different beans in a conformational ensemble. So same sort of concepts can apply here to calculate the free energy change, right? Here the fluorescent or the fraction of the proteins uh, at this particular sort of state where the fluorescent intensity is 256. So we can calculate the fraction unbound here. So that is sort of the formula. The formula is like this basically. So y of f minus y divided by y of f minus y of u. So in this case, the y of f is the folded state's fluorescent intensity minus y is basically at that particular state what intensity is being indicated. You can divide that by yf minus yu, so folded minus unfolded value. If you do that, you will get a value of 0.349. So that will be the fraction of the unfolded, right? If you want to get the fraction of the folded at that particular state, you can subtract this value from 1, you will get 0.651. Now, the particular formula that I was talking about, so this delta G formula, which will be indicating the free energy change from that particular free energy change of that particular state from the initial folded state. So it can be indicated by minus RT ln K. And in this case, since it is basically asking in comparison or with reference to the initial folded state, so we will use the fraction of folded as the denominator here. So it will be minus RT ln of FU divided by F of F, which if you basically calculate it out, so the R is basically 1.987. So the unit for the R here is calorie per mole per Kelvin. You multiply that by temperature value 300 Kelvin. So the Kelvin that is basically crossed out. And then this logarithm ln of 0.349 divided by 0.651. So this in total will give you a value of calorie per mole, right? So if you calculate it out, it will be 371.631 calorie per mole. Uh, in general, for the free energy changes, we use a kilocal per mole for unit. So you can convert this to kilocal per mole by dividing this with 1000, right? So that is sort of the concept here. So I will once again share the other screen. I think the modeling has completed there. So just take a look. Okay, so hopefully that is visible now. Now, in this particular case, so let me just unselect everything here. So one new tab has popped up after the modeling job was finished, the loop modeling job in Modeler. And what we can basically see here, we can see 5 different models. So what we give the number of value for the number of models to be generated. So that was 5. So here we are getting hashtag 1.1 to hashtag 1.5. We are getting some corresponding Z dope score as well, right? So the Z dope is basically the normalized dope score, which is indicative of what is the quality of the new model that we have gotten. And the more negative value, the better it is. So firstly, let's see what where the models, what do the models look like in these particular regions where the structure was missing. Just one moment. We can open this up on the side. And what we can see on the left hand side is the particular models. Now you will notice if you press, for example, hashtag 1.1, of course you will see the blue colored basically model that has been generated in the place of these dotted lines. So that is indicative of the model conformation for this region, the loop model conformation for this region. For example, in this one, it is clearly visible. So this gray one was our initial structure, the unmodeled structure, but the dotted lines are present. Now, using the sequence information, it has generated a structure for this missing missing residue range, right? So this is the conformation what it is looking like. You can go through the other ones. For example, in hashtag 1.2, the structure is of course slightly different. Hashtag 1.3, the structure is slightly different. Then 1.4 and 1.5 you will see slightly different. So among all of these, if we have to take a structure, it is best to take the structure 5 here, hashtag 1.5, which has a z-dope score of minus 0.74. So the more negative the score, the better the model in this case, right? What we can do is we can just save the PDB from the file option and then the save PDB option, and then it will ask which models you are trying to save. You have to go for a single file, and then you can press this one, and then for example 1.5 this one, and then you can save. You can open up the structure and it will be modeled basically. So that is how you can perform loop modeling for missing residues using Chimera and the associated plugin for Modeler. So you have to separately install Modeler. You have to get, for example, license key of Modeler. If you provide your academic institutions, they will provide the license key. You can install that Modeler software. Then you can use the path of the executable for Modeler. You can put that in the model tab for the or the minimize. It was I think model/minimize loops tab, something like that, right? Yeah, in the structure editing, model/refined loops. So this tab you can put that path, and then you will get the model structures here after doing the modeling. Okay, so in our
Here, I just want to refresh.
uh,
So initially, why there was the dotted line? Why initially that was not modeled?
so this particular structure is from a PDB. It is basically taken from a PDB database. So what is happening is that when the particular research group crystallized this protein, so for these regions, this dotted line, these regions, due to very high flexibility of the residues being present here, they could not properly capture what could be the exact uh, basically coordinates of the residues that are present for each atom in this residue range. Okay, so that is why, since it could not be captured due to high flexibility, so that is why the dotted lines are present there in the initial structure. So for the every structured part where the secondary structures are present, so those are not that highly flexible. So their corresponding coordinate values for those atoms could be captured. So that are present in the PDB structure itself. But for the dotted line, in the crystallization process itself, the particular conformation of those loops could not be captured essentially. So that is why if you want to study using this PDB structure, you have to first model the loop regions to initiate this study. So that is why we are doing a loop modeling for that bit. Okay, for those missing regions.
Because of the flexibility of those atoms, they were not captured.
Yeah. You can think of it that way. So in the crystallization process itself, through X-ray crystallography, the proper coordinates could not be captured. Okay.
So now when you modeled this, so since they were not modeled initially, so
Yes.
now, I mean, what did you do, or what is the mechanism behind, on what basis were these modeled? Like you showed just 5 structures and you selected one of 5. I got more negative value, so you selected that. So now when initially the group that they have done, so they could not crystallize that
Yeah.
structure, so that's why it was dotted lines. So now when you modeled, so what it has taken, because here you are not doing any crystallization study or anything, so how the system has modeled that this structure, the dotted lines?
Yeah. So in our slides also, if we go through that, so in the entire process of homology modeling, you would have seen that a loop modeling portion was also present in that, right? So in the loop modeling, what basically the protocol was doing, that whenever there was a gap in the alignment, for example, in homology modeling, so what it was doing, it was either searching the PDB database for similar sort of sequences in some other proteins, so that it can take the structure from there, or it was using the ab initio protocols. So the ab initio protocol was based on the first principles, whether we can get the energy minimized structures for what the backbone should look like. So those kinds of processes are being followed in this loop modeling. So for the ab initio modeling, it can take a lot longer basically. Now, this has only generated a confirmation through a process that is not that computationally expensive. So you can expect that the accuracy could be slightly lower, which is also represented in this Z-dope score also minus 0.74. So if you take a look at this particular tab from the group that built the modeler software, we'll see that scores lower than -1 are being expressed as native-like structures. So in our case, the model was not having quite a native-like structure because, of course, the loop region was in a large number of residues are present. So the proper, for example, 3D orientations, the larger the number is, so there will be more possibilities for the sidechain orientations to be present. So those regions will be highly dynamic, and it has only captured with a Z-dove score of minus 0.74 as the best one. So in any way, you have to do further, let's say, post processing, you can say, or further equilibrate the structure to find out where the exact minimal lies, sort of, through some simulation based studies.
Okay.
So for this, okay, yeah. For the Swiss model tab also, we are seeing some results here. So in this particular case, with the basically available hits that were present through the research database and then after alignment and so on. So we are seeing that the different results or the outputs have been given. So in this particular case, only model 01 and model 2 are being given here. So that means only 2 hits it could identify with their particular GMQE score, which is a score for evaluating the models being present here. Also, you'll notice that some sort of color code is being present in the 3D structures. So yeah, so I guess in this case, the color code for the color code, see the blue colored are being indicative of the core region of the protein, which was being able to model correctly with a high sort of confidence level. And these orange colors you will notice is mostly the loop regions. So for the loop regions, again, they are highly flexible. A number of conformational orientations can be present there. So the proper folding conformation for these sort of stages, it can vary basically. So a high confidence level is sort of not possible for these loop regions in a lot of cases, right? Then you can compare the sequence identity. For example, here 93.39% identity was there and so on. You can compare your different results. Let's say in a particular tab, you are seeing model 1 to model 10 different models. Then you can compare the parameters, the GMQ scores, the sequence identity, what template was used in the modeling. You can compare all of that and take a proper sort of model structure for your further studies. Okay. So I will just share the slides again. So that was basically some concepts of homology modeling. Here we had the urea denaturation experiment. Now here is one interesting question. What does CASP or CASP stand for? Now, in the context of protein modeling or protein structure prediction, these sort of things, CASP is basically a very highly regarded sort of assessment competition, you can say. So does anybody know what CASP particularly stands for from these options? So CASP essentially stands for Critical Assessment of Protein Structure Prediction. So it is a widely regarded competition for the assessment of different algorithms that can predict the protein structures. Okay. And of course, we know that the history of CASP. So CASP was basically started by Professor John Molt from University of Maryland, and then his colleagues. They started this competition because anyway, so a lot of people were trying their hands at the protein folding problem, which is basically that the number of ways that a particular protein and its side chain orientation and so on can fold is pretty numerous, right? So through brute force calculations and so on, this kind of values or particular conformations for the folded proteins was kind of hard to get. So Professor John Molt and his colleagues, they started this CASP competition, basically the critical assessment of protein structure prediction, in a way that will allow different research groups who are sort of trying to solve the protein folding problem uh, to assess their algorithms in a bi-yearly competition, basically, right? Now, we know that, for example, in CASP, uh, from 1 to CASP 12, uh, so in those cases from 1994 until the CASP 12, it is a bi-yearly competition. So in that range, uh, although a lot of different research groups are trying to devise an algorithm that could predict the protein structures properly, so they could not reach the threshold value of 90. So in 90, from 1 to 100, the scoring system basically worked in this protein structure prediction assessment. And if any algorithm, if it was able to reach the score of 90, then okay, you can say that it is very confidently solving the protein structure. And what the organizers would do is basically that from a newly crystallized protein, so it is crystallized through experimental procedures, right, extracrystallography and so on, and it is unpublished. So no other protein structure for this particular protein is present in the PDB database. It is an unpublished data. So what the organizers would do is that for all of the participants, they would just provide the sequence and they would ask them, okay, you predict the structures and you give the output. And then they will match their already crystallized structure with the predicted ones. And then they will identify, okay, with how much accuracy the prediction algorithm was working. Now, even though they set a threshold score of 90, so from a large, basically throughout the large number of years, so right until CASP 14, so nobody could achieve this threshold of 90 score for their prediction models. So in CASP 13 in 2018, what happened is that one group they decided to take their algorithm to this competition. And even though they could not achieve the threshold score of 90, they were already better at the prediction than the other models that were available at the time. So that was a jump from the already available models, but still they could not achieve the score of 90 in CASP 13. So basically the model was still not perfect. However, after they modified their models, they did some tweaks, they worked on it more. And then after the at the next CASP competition, so it was in 2020, so this group again put their modified version of the algorithm to the test. And for the first time in the history of the CASP competition, their algorithm could achieve the threshold score of 90 for the prediction accuracy for the structure that they were given, right? So this is at the time that people are talking about, okay, the protein folding problem is probably solved now, because one algorithm or one program can now predict the structure of a newly found sequence, basically, right? So does anybody know what is the name of this program, what group I am talking about, research group that devised the program is, which group, and what software, basically? So it was, of course, the very well-known AlphaFold, right? So it
No,
is,
sir.
yeah. So it is written here, the success of AlphaFold 2 in CASP 14. So CASP 14 is basically the competition in 2020 when AlphaFold became the first prediction model to predict the structure of a unknown protein from a sequence, basically of a protein with unknown structure from a sequence, specifically with their threshold score of 90. And as we all know, uh, the Nobel Prize in Chemistry for 2024. So one half was awarded to Professor David Baker from University of Washington for computational protein design, and the other half of the Nobel Prize was jointly awarded to two researchers from DeepMind, right? So one is Demis Hassabis, of course. He is the CEO of DeepMind, and one is John Jumper, who led the AlphaFold project, basically. So they were jointly awarded the other half of the Nobel Prize in Chemistry in 2024 for protein structure prediction, and basically for the development of AlphaFold, AlphaFold 2, and so on, right? So if you know the history of the AI research of DeepMind, you will know that their earlier research kind of started with not with in the protein folding prediction or protein structure prediction, but in solving some of the board games that are quite widely popular, right? Their first sort of AI program, which was named as AlphaGo, so it was a program that could that ultimately beat some of the best Go players. Go is a board game, so Go players in this game. So some of the highly regarded Go players, some of the highly rated among all. So it could beat those. So Go is basically kind of like chess, but it has a lot of large board, and a lot of time. For a lot of time, it was considered that okay, no computer program could probably able to beat a human Go player at the highest level ever, basically, because Go is kind of considered a more intuitive or positional kind of play in a sense. So brute forcing calculation does not work always here. Uh, Now the intuition thing for that, the DeepMind team for the AlphaGo, they sort of tried their hand at solving the Go problem. You can say in a way. So they could see, they were trying to see if their newly devised program could sort of learn by playing against itself one after one after one turn, and then after like thousands and lakhs of turns, or that it could improve from being a novice to be at the highest level that a human can possibly reach, and then can it go beyond that? And they quite successfully did that. So there is a very good documentary on YouTube. You can search it up. So it's called AlphaGo the movie, which kind of goes through the entire process of kind of building the AlphaGo and its matches against the rated Go players and the legendary Lee Sedol. So those kind of are those processes and those things are sort of documented there. And then after that, they tried their hand at solving chess also. So they also built Alpha Zero, which is their chess playing program, artificial intelligence program. And then after that, they tried their hand at protein folding with the AlphaFold project, DeepMind. They tried. So that was basically the origin of AlphaFold and how it was able to predict the protein structure with very high accuracy. The other part for the Nobel Prize went to Professor David Baker for computational protein design. So his idea was that can we devise can we make, for example, a new enzyme or a new protein that is not present in nature? Can we tailor that to a particular sort of function, let's say? So let's say there is a target binding site. So can we make an enzyme that will bind to that target binding site for which there is no known enzyme that is available, that is known in the nature? And then after that, if we sort of predict the 3D conformation that the enzyme needs to be present for this binding to occur, let's say, then can we predict what the sequence will be? Of course, we can do that from the structure. So what kind of sequence would ultimately give rise to this sort of structure? And then what sort of genetic code needs to be there for this sequence to be formed? And then those genetic sequences can be expressed in some bacterial systems. And then you are getting a new protein that was not earlier present in the nature, that is tailored for particular functions, right? So as Professor David Baker says, like in a lot of his speech, so if you for a lot of problems that we see nowadays, for example, environmental sustainability problems, a lot of like drug and medicine related problems and so on. So probably if we let evolution course through these problems, some sort of mechanisms will be devised by nature inside us to combat some of these problems, right? But evolutionary processes take a large, basically amount of time. So instead, can we tailor these new proteins or these new enzymes ourselves, which will be performing particular functions, which will be more sophisticated, uh, and tailored to some of the functions that is kind of at the heart of the problems that we see now. For example, can we make one enzyme that could, let's say, degrade plastic? So that is also one of the problems that you mentioned. So different types of problems in medicine, in environmental sustainability, in new technologies, Professor David Baker's lab basically work on. So I have given link to several videos here. So I think the left two are sort of general videos about the Nobel Prize and the associated protein structure prediction and computational protein design. The right hand side, I believe, are their basically Nobel Prize reception speech, which they gave. So you might take a look at all of these videos. I'll share the slides, of course. So yeah. So another thing that I was kind of wanting to mention that Professor David Baker's lab, they also devised something called Rosetta, which is again also a folding software. And AlphaFold's initial sort of training was done using something called Fold It. So Fold It was a game basically that was again devised by Professor David Baker's lab, which was distributed among different people. So different gamers, they tried their hand at this gamified sort of protein folding game, and they were able to achieve some good results. So the AlphaFold's initial training, I think in one of those videos I saw that their initial training was based on can it solve the this protein folding game, can it play this game perfectly. So that's how they transitioned from doing AI research for the board games like Go and Chess to particularly this protein folding problems, which again Demis Hassabis he was very interested in for a long time. So yeah, you can take a look at these videos in the shared slides. And then I have again given link to the two different papers for Professor David Baker's lab and then the AlphaFold team as well. So for example, the left hand side is mentioning scaffolding protein functional sites using deep learning. And the right hand side is basically the paper for AlphaFold 3. So you can take a look at those to gain maybe more insight into their projects. For today, I think I will wrap up the classes. And the next set of concepts I will discuss in the next class, and for the next week's contents again, we will discuss in the next classes. Okay. So thank you everyone for joining. You can go through the videos that are being linked in the slides. Again, please go through Professor Pumiha's lectures carefully because a lot of questions related to the assignments will come from his lectures, and try to go through the problem solving sessions for the other TAs. And again, we'll meet in the next class. So again, thank you everyone for joining, and we'll discuss the further concepts in the next class. Yeah.
Excuse me, sir.
Yeah, please.
I'm getting the link of only this meeting. I mean, your meeting. You said other TIs.
So you have joined the course, right? So I think from the NPTEL platform, you should be receiving links for the other TA classes as well. So one email from online courses at nptel.iitm.ac.in. They basically provide the links for all 3 TA classes to every learner that are registered for these courses. So you are not getting those links?
No, sir. I'm getting only your meeting Google links. That's it.
Okay. I will basically provide the link in the chat maybe. Just wait a minute. So this is one class that goes on in Saturdays. Hopefully you can note the link here.
Yes, sir.
Yeah, so that is regularly on Saturdays for week 1 to week 12. And let's find it for Monday.
So this isn't yours.
No, this is in mine. So mine is on Friday. So that is the Saturday class. The same link will be for all of these classes. And then the second one, what I have given here, it is for the Monday class. So the time is also mentioned here. Wait, I think the regular time I have to check whether it is this one or just a minute. Because I think in the last class, some schedule change was there. So just wait a moment. I'll give the link for some of the earlier classes actually.
We can attend either of the 3 classes, na? Or all
Yeah, yeah.
three needs to be attended?
No, no, no. So the attendance is basically whenever time you find preferable, so you can attend those classes. All of the classes will be the recording will be given through YouTube links. That's fine, but you can attend any or all of the classes based on your preference.
Yeah, that's what I am attending. Sometimes your class or sometimes Ramakrishna class.
Yeah,
So I just wanted to know I can attend either, na, or the same content will be there, right?
yeah, exactly. The same content will be there. So just one moment, I will. The same content will be there. The same sort of core concepts will be discussed by all three TAs for each week. Okay. So there should not be any problem in that case.
But the questions will be different, right?
Yeah. So it will be kind of taken by different TAs. So their slides and their approach might be different. The questions might be different based on what they have put in their slides and so on. So that is for Friday. Uh, yeah. So I believe these should be the two links. So on Saturday, I guess it is from 4 to 6. So can somebody confirm whether the Saturday's class is from 4 to 6 or not? And whether this is the correct link?
Saturday is 4 to 6. Even though I didn't attend any of the Saturday classes, but
Uh-huh.
I'm getting email notification for 4 to 6.
Yeah.
But
Yeah.
last time I think it was not there.
I think last week basically there was some change in schedule, I think. So
Okay. Otherwise, it is 4 to 6.
yeah. And the Monday's class is from 6 to 8, as it is mentioned here. I think there should be a shared spreadsheet somewhere. I'll just take a look. Just one moment. So I'll probably just share the link to the spreadsheet also in the chat. Problem solving sessions. So this document basically should lead you to the shared spreadsheet where you can get all of the recording links and so on. Yeah, I think it works. Okay. And these should be the two classes for Monday 6 to 8. So you can go to this link, and then for Saturday 4 to 6 you can go through this link for the classes. And I think if you are not getting all of the notifications, so what you can do, you can post the post the concern on the forum page or the discussion page, and hopefully the admins they will take a look whether notification was off in your case or something like that. Okay. Or alternatively, you can
Okay.
also probably email the NPTEL team if an email address is given. So they will fix
Yeah.
it.
Sure.
Yeah.
Sure, sir.
Okay. Okay. So again, thank you everyone for joining today's class. And again, we'll meet at the next Friday's class. Okay. Thank you.
Thank you, sir.